Is it clear? So kidney as a large organ is not producing. It is a collection of such units which are called as the nephrons. Now these nephrons are structural and functional. Functional means the actual function of urine formation is carried out by nephron. Is it clear? So let us understand how is the structure of nephron and how it helps for the process of urine formation. Clear? Okay. So let us draw the structure of nephron. Is the board visible? Children? Is it visible? Okay. So have you ever seen this drawing, children?
Yes, ma'am.
What is this drawing? Tell me.
Nephron.
This is the nephron. Okay. So you have seen in 10th standard, it seems, right?
Yes, ma'am.
So you have seen in 10th standard. Now you will tell me what are these parts. So I will just label, you will tell me what is this part. What is this part?
Afferent arteriole. Glomerulus.
It is glomerulus. What is this part?
Bowman's capsule. Bowman's capsule, ma'am.
Ans capsule. Okay. And combinedly, what these two are called?
Malpighian body.
Malpighian body. Okay. These are called as Malpighian body. Okay. Here. Oh, what is this part? This one. You know about this? This is proximal
Convoluted tubule, ma'am.
convoluted tubule. And what is this part?
Distal convoluted tubule.
It is distal convoluted tubule. Right? Okay. Now, what is this part? The U-shaped part. This blue color U-shaped tube. What is that?
Loop of
That is called loop of Henle.
Henley.
Okay, loop of Henle. And what is this part? This branches of blood capillaries are called.
Renal
They are called as the
capillary.
vasa recta. They're called as vasa recta. And they are branch of this renal artery. Okay, so this is vasa recta. What is this tube called? What is this tube called? This one. Where the nephron is joining. What is that part? This one.
Open to the.
It is collecting duct. Collecting duct. Okay. Is it visible or not?
Yes, ma'am.
Collecting duct. Right. Okay. So, this part, this branch, glomerulus has one entry point of the blood vessels and one exit tube also. So, the exit tube is called as efferent arteriole, and this part, the entry tube, I'm just writing here, this is not the correct way of labeling. As space is not there, I'm writing here. This is afferent arteriole. Okay, this is afferent arteriole. The arteriole which enters and forms the glomerulus inside the Bowman's capsule is called afferent. And after the glomerulus part is formed, it again goes out as another arteriole that is called as efferent arteriole. Here, and in the middle there is a tiny ball of blood capillaries that is called as our glomerulus. Is it clear? Okay, right. So glomerulus is held inside a cup-shaped structure that is called as Bowman's capsule. Clear? Okay. So let us go with details of the structures of nephron. So this loop of Henle, actually, this part of loop of Henle is called is it visible? This part is called the descending limb. Okay, descending limb of loop of Henle. And this one, this particular one is called, it is called as the ascending limb of loop of Henle. Okay, ascending limb. So which one is ascending limb? This part, which continues as the distal convoluted tubule. And the part which continues after the proximal convoluted tubule is called as the descending limb. So the tube starts from here, descends down, again goes up, becomes convoluted, maybe, or coiled in this part, and it joins into the collecting duct. Is it clear? Okay. So this part, this is one structural unit, and this collecting duct can be attached with many nephrons together. Okay, that is why these points I have drawn like this. So this is one nephron attached here. Here one more can attach. Here one more. Here. So one collecting duct Many nephrons can attach and send the filtrate finally. Clear? So what is this part? So the nephron has few important parts. One is the Malpighian body part, the topmost part, right? Next comes the tubular part and then the collecting duct. It joins into the collecting duct. The tubular part has three regions. What are the three regions? One is the proximal convoluted. Convoluted means coiled structures. Yeah? So convoluted is coiled. So the proximal means the towards the starting point. So towards the starting point, whatever convoluted part is there, coiled part is there, that is called as the proximal convoluted tubule. Clear? And there is a U-pin-like structure. After this part, the proximal convoluted tubule part, there is a U-pin-like structure. Okay? So what is this U-pin-like structure called? It is called as loop of Henle because it forms a loop inside the medullary area. Is it clear? So this is called as loop of Henle. Now loop of Henle has two arms, right? One is the descending limb and the ascending limb. So the tube comes down, right? Again moves up. So this is descending limb, this is ascending limb. This side is ascending, this side is descending. And after that, again one convoluted part is there. What is this convoluted part called? This is called as our distal convoluted tubule. And finally, it enters into the collecting duct. Clear? Now coming to the blood vessel. Now which blood vessel will come near the nephron? Nephron gets its blood supply, the blood supply from the renal, the branch of renal artery. Clear? So as nephron is a very tiny structure, so when artery enters, the renal artery enters into the kidney, the renal artery cannot go near all the nephrons. So it has to make branches, like we have already studied. The blood vessels make smaller branches to reach different corners of one organ. And finally, they form capillaries to reach each and every cell. Clear? Similarly, this particular blood vessel is a branch of renal artery. So, it is named as the efferent arteriole. Right? So, this is a branch of renal artery. It enters into this Bowman's capsule. Right? Enters means what? Its location is there. This blood vessel forms a knot-like structure. Suppose you are holding a thread, right? And you are just in finger, you rotate sometime and remove it and make a ball of thread in between the total length of thread. So it is just like that. Suppose you're holding a big length of thread, right? In the middle, you make a ball of thread. And both the sides are straight. This structure is just like that. It is straight coming towards the nephron. Inside the Bowman's capsule, they are coiled. Again, when the blood vessel goes out, it is straightened as a efferent arteriole. You are able to understand what I'm telling? You are able to understand the blood vessel comes straight near the Bowman's capsule, coils heavily, okay? And then after when it going out of this place, Bowman's capsule, when it is the location moving out, when the blood vessel is here, it is straightened again. The knot is made only here and it has a very big purpose, right? This particular part has a very big purpose. What is the purpose? The purpose is to filter the blood, to prepare the filtrate and send all the filtrate into the tubular part. Okay, so the filtrate is going to get squeezed from the blood capillaries here, and then it should be entering into the Bowman's capsule, and then it has to move inside this tubes. Okay, inside this tubes. You are able to understand? You are able to understand what I'm telling?
Yes, ma'am.
Okay. Now this part is completed or not? Shall I erase it or not? Shall I erase it? One more thing. Am I audible? Children, am I audible?
Yes, ma'am.
Okay. So one more thing I forgot about after this efferent arteriole comes, it starts coiling around the tubular part of the nephron. Okay, the efferent arteriole makes branches and completely coil around the tubular part, right? And that is known as the vasa recta. The purpose of this is later we'll be discussing. First, the filtrate will come, then again, few of the things have to go back to blood. So, for that purpose, we need a blood vessel very close to the tubular part. And that is the reason this vasa recta is present, coiling very close to the tubular part of the nephron. Shall I erase this?
Yes, ma'am.
Have you all
Yes, ma'am.
put it down? Okay.
Yes, madam.
Ashwini, very excited today.
No, madam.
Why you are not feeling well? Huh? By the way, happy Navratri to all of you. I forgot to inform.
Thank you, madam. Wish you the same, madam.
Yes, ma'am.
For tomorrow.
Madam, today I'm fasting, madam. That's why I'm not excited.
That's right. Otherwise, you would have been shouting more.
Yes, madam.
For what reason you are fasting today? Because of Navratri? Yashashwini?
Madam,
For what reason you are fasting today?
yes, madam. Today Saturday, na, madam.
Okay. Okay, let us go to the next part, that is urine formation. Urine formation. Okay, urine formation means what? We studied the structure of nephron, right? So, what is the function of nephron? Nephron's function is to produce the urine,
Yes,
right?
ma'am.
The production of urine, the filtrate, after all the process is completed, the final filtrate, which has to enter into the collecting duct and to be removed out, that is called as the urine. Starting itself, whatever filtrate comes inside the Bowman's capsule are not called as urine. Clear? What they will be called? They will be called only filtrate. Finally, after all processes are completed, then only the final product which is removed is called as urine. Yeah, so there is always a difference between the amount of the amount of filtrate and the amount of urine produced. Yeah, okay. So the urine formation is completed in 3 stages. Okay, what are the 3 stages? The glomerular,
Filtration.
filtration,
Tubular secretion.
glomerular reabsorption, selective reabsorption, okay, and the third phase will be tubular secretion. Okay. But what happens in the majority of mammals, right? Majority of the mammals or vertebrates, we can say, tubular secretion doesn't play much of the role. It is playing minor role. Only in organisms who are like desert organisms, in them tubular secretion plays a major role. Like they want to they store more water, they want to retain more water. In such organisms, the tubular secretion plays major role. But for organisms like mammals who are not in coming that category, they have only this and this as major part. Okay, glomerular filtration and selective reabsorption. So tubular secretion for us is very small, minor process. Yeah. So let us first go to the glomerular filtration. Now, glomerular filtration means what? What do you understand by glomerular filtration? Anybody can answer? What is glomerular filtration? The filtration done by glomerulus, correct? The filtration carried out by glomerulus is called glomerular filtration, right? On an average, how much blood is filtered by the kidney per minute? Can anybody say? It is 1,100 to 1,200 ml.
200.
Okay, 1,100 to 1,200 ml of blood, which is filtered by the kidney per minute. Okay, then the filtration is done in the glomerular part, glomerular and Bowman's capsule. That means the Malpighian body. So let me erase this part. Then we'll continue with glomerular filtration. So this process is also known as the ultra filtration. Okay. This process is also known as ultra filtration. Now, why we call this as ultra filtration? Yeah. So the thing is, what exactly gets filtered in the glomerulus? When the glomerulus, suppose if I draw just the Bowman's capsule part, okay, this is the Bowman's capsule, right? So in this area, this is the blood vessel. Okay, so this is Malpighian body. Now this is efferent arteriole, this is efferent. So I'll write A and E. Okay, how exactly the ultrafiltration takes? So whatever filtrate now will come out, they are called as glomerular filtrate. So the filtrate which is coming out or which enters into the tubular part is called the glomerular filtrate. Right? The glomerular filtrate. Now the question is, filtrate is coming into the tubular part, that we all know. Now what mechanism is there that the filtrate is squeezed out from the blood vessel? Why the filtrate is coming out? Now what is contained in the filtrate? The glomerular filtrate contains plasma, okay, and it also contains amino nutrients, water, Okay, minerals, ions. These are few substances which are squeezed out. Now, how they will be squeezed out? Because there must be some pressure generated so that the blood passing through this capillaries will squeezed out. Suppose you think you are holding a cloth and inside the cloth, cotton cloth, you are having some liquid, fluid structure. Here. So suppose you squeeze that cloth, then what will happen? Through the pores of the cloth, the fluid will move out, will try to move out. Same thing is happening in the glomerular. When pressure is exerted on the glomerular this area, when anybody wants to say anything? Anyone wants to speak? Okay, so when the pressure is exerted on the blood vessels in the glomerular area, the blood vessel pores, the wall has, and those pores will squeeze out the majority of the plasma, water, minerals, many hormone molecules, many substances will come out in the form of a fluid which is called as the filtrate. Right? So what else comes out? The main substance, that is the nitrogenous waste. Okay, the nitrogenous waste also comes out along with this substance. Clear? So now thing is how this pressure is created. Now when we think of this arteriole has a larger diameter in comparison to efferent arteriole. So I'll just write here efferent arteriole has bigger diameter than efferent. Okay, you're able to understand, you're able to hear? Am I audible to you all?
Yes, ma'am.
Okay, so what I said just now, efferent arterioles are having bigger diameter than efferent arterioles. Which is efferent? The arteriole which goes out from the glomerulus is efferent. Efferent is the side which enters. That means the blood flows with the force, but it cannot come out through the with the same force. Why? Because the tube diameter is lesser this side. The diameter of this arteriole is less in compared to this. So blood will flow faster, but it cannot come out of glomerulus faster. So it will be lagging, right? So you just think water is entering into a tank or any area with a bigger tube and coming out with a very tiny small tube. Then what will happen? The water will be locked there. The water will be contained there for some time and slowly it will be going out. So when more and more blood are contained here, then what else is going to happen? It will exert pressure. Clear? The fluid is increasing inside the tubes here. So that will exert wall pressure. Is it clear or not? The fluid means the blood which is moving through this capillaries. So when the blood cannot flow freely, it will be collected in the glomerular area. And second point, why the pressure is exerted. The first reason why pressure is exerted is this. So this is reason number 1. You're able to see here? You're able to see the board? This is our reason number 1. Efferent arteriole has bigger diameter than efferent. Now, what is the reason number 2? Reason number 2 is the coiled glomerulus slows down the flow of blood. The coiled glomerulus. Suppose if I draw here, this is one blood tube, okay, and this is another blood tube. Okay, tell me where the blood flow will be faster. Which tube the blood flow? This is tube A, this is tube B. Which tube the blood flow will be faster? Which tube the blood flow will be faster?
Ma'am, A. A, ma'am.
It's tube A. Why? Why does tube A?
Straight.
It is straight. The tube is straight. So there is no obstruction for the blood to flow. Suppose it is the tube B, then it is a coiled tube. So it will take time to move in this tube. That is what is happening here. If the glomerulus would have been a straight tube like this, then the blood would never have created pressure on the capillary wall, and never we would have been having a functional kidney. It is because of glomerulus the kidney is able to produce the filtrate. If it would not have been a coiled structure like this, straight like this, then we would not have got the filtrate. So two reasons for the pressure which is created in the glomerulus. What are the two reasons? First reason, afferent has larger diameter than efferent arteriole. Clear? Second is the coiled glomerulus slows down the flow of blood. So both combinedly work together to create a pressurized condition in the glomerulus, and that squeezes out the plasma from the capillaries. So what remains in the blood? Little amount of plasma, RBC, WBC, all those cells, larger proteins. So whichever one is the larger molecule that cannot pass through the capillary wall, only the smaller molecules, water, plasma, that will come out. But the larger molecule remains as it is in the blood. Here, and the blood again becomes the vasa recta. The capillary becomes vasa recta. But in this condition, what happens? Only nitrogenous waste has to be removed. So these are our required substances. Here, so this required substances has to be sent back to the blood. That will be our selective reabsorption. So selective reabsorption takes place for plasma, nutrients, water, minerals, ions. That will be selective reabsorption. Now what should be there remained in the filtrate? That is nitrogenous waste, some drugs, some amount of water, that should be discarded from the body. Is it clear?
Yes, ma'am.
Now let us go for glomerular filtrate. So what is the quantity of glomerular filtrate, and what are the different layers that the glomerular filtrate crosses to reach the tubular part? I'm going to erase this side and I'll leave just the glomerulus. So let us write down the three layers. What are the three layers? Okay. So the three layers which the filtrate crosses, right? So first is the endothelium of blood vessels. The three layers. Layer number one, endothelium of the blood vessels, glomerular blood vessels, glomerular blood vessels. Second layer that crosses is the epithelium of, no, actually two layers. The epithelium of Bowman's capsule. This and this. And one basement membrane. Actually, one basement membrane will be there. 3 only. This is basement. Yeah, I forgot about this. A basement membrane between the blood vessel and Bowman's capsule will be there. Right? So second one is basement membrane. Basement membrane. Third one is the epithelium of Bowman's capsule. Okay, so these are the 3 layers the filtrate has to cross. Is it clear? So what is that? First is endothelium of blood vessel, glomerular blood vessel. So blood vessel is like this, capillary, right? It has single layers. You're able to understand? So this is the endothelium. Endothelium of blood vessels. Is it clear? Then what happens? There is a basement membrane, packing tissue. The basement membrane that has to cross the filtrate. That means the plasma here that will come out, crossing the endothelium, basement membrane. Then comes the epithelium of Bowman's capsule. Okay, so it has to cross all this. Then the tubular part comes. Clear? What happens here actually in the epithelium of the Bowman's capsule? Okay, the epithelium of Bowman's capsule, there are some very special cells. Okay, so special cells in the epithelium. I will write in black. Just a second. The special cells called podocytes. Okay, what are the special cells called? They're called as the podocytes. Right? The podocytes are arranged in an intricate manner. Right? In a very complicated manner so that they leave minute spaces. That is called as the filtration slits or slit pores. They are arranged like, suppose if I show it properly, podocytes will be arranged like this. They are not regular, they are intricate, complicatedly arranged. These are the cells. Okay, so here what happens, tiny pores will be there, gaps will be there. Okay, these gaps will take all the filtrate. So you can see the gaps. The gaps are made because of the arrangement of podocytes. Okay, so tiny pores will be there on the Bowman's capsule. So what are these tiny pores? Tiny pores called filtration slit, or they're also called as the slit pores. Slit pores. These tiny pores are called filtration slit or called as slit pores. They are the reason for the filtrate to come into the tubular part of the nephron. So this is the Bowman's capsule, right? The Bowman's capsule, the glomerulus is there. So it has the podocyte which makes gaps in between the cells so that the filtrate leaks out into the tubular part of the nephron. Clear? Is it clear?
Yes, ma'am.
Okay.
Yes,
Now
madam.
blood is filtered so finely through these membranes that almost all the constituents of plasma will come out except the larger molecules. Yeah? What are the larger molecules? The proteins, the larger cells, all those things will remain in the blood vessel. But what will come out is the nutrients, the plasma, the water, mineral ions, all those things will come out into the tubular part, right? So as the filtration happens in a very minute level, till minute molecules, all the molecules that come into this, so as ultrafiltration process, right? So maximum filtration happens here. That's why it is ultrafiltration. Ultra means what? To the highest level, right? To the highest level, ultrafiltration. The like maximum, how much possible, that much filtration will take place. So it is called as ultrafiltration. Only few molecules are left in the blood. Is it clear? Okay.
Yes. Yes, ma'am. Yeah, ma'am.
Read this. So next is glomerular filtration rate. Okay, GFR. Now, what is GFR? Amount of filtrate formed in the kidney. Amount of filtrate formed in the per minute. Amount of filtrate produced or formed in the kidney per minute is called as glomerular filtration rate, right? So it is 125 ml per minute. 125 ml per minute. That means 180 liters per day. Yeah. 180 liters per day. But this is not our urine formation count. Remember that. This is only amount of filtrate. That means the filtrate which comes from the blood vessel enters into the nephron, not the filtrate which is removed from our body. We would have been dying if we remove 180 liters of the water or urine from our body per day. Yeah. So actually, the urine which is released per day is 1.5 liter per day. Yeah. The urine which is released from normal human body is 1.5 liters per day. Okay. This is liter per day. But how much filtrate is coming? Filtrate is 180 liters. That means
180 liters per day.
9% filtrate is selectively reabsorbed. Okay, 99% of filtrate is selectively reabsorbed. If it is 1.5 liter per day, but filtrate is 180 liter, 99.9 also we can say. That is reabsorbed. Okay, so very less amount is filtered. 99.5 or something, that will be reabsorbed. Okay. Now, the kidney has a built-in mechanism for the regulation of glomerular filtrate rate. So what happens? The mechanism is carried out by juxtaglomerular apparatus. Shall I erase this part?
Yes, ma'am.
Have you all written this one? Have you all written?
Yes, madam.
Okay. So what happens? The glomerular filtrate rate, the kidney has its own mechanism to control the glomerular filtrate rate. So who controls in the kidney? There is a juxta glomerular glomerular apparatus, juxtaglomerular apparatus, which is known as JGA. Okay, where it is present, this is present, very sensitive region formed by the cellular modification on the distal convoluted tubule and afferent arteriole. Okay, as a location of their contact. So where this part, these are special tissues on BCT and the afferent arteriole. Okay, afferent arteriole, BCT and afferent arteriole. So that is the location for this juxtaglomerular apparatus. Special tissue which are very sensitive for the amount of filtrate which is formed. So when more filtrate is formed, they will activate so that the filtrate rate will be, they will get activated, and one hormone they release that later also we are going to discuss the control of urine formation of kidney function, that we will be reading later. Yeah. So how they actually work, again we'll be reading a later part where the kidney function, how kidney function is controlled. In that part, we'll be reading again this thing, right? How it is controlling. The thing is, when GFR falls down, the amount of GFR, that is glomerular filtration rate, it will, when it falls down below its normal level, then it will activate the juxtaglomerular cells to release a hormone, which is called as renin, right? So this cells, I will use the pen just a second. This cells release renin, a hormone. Just a second. Which increases the glomerular blood flow, increases the glomerular blood flow, and the GFR. Okay, glomerular blood flow and the GFR. So actually, this is a Is it visible, children?
Yes, ma'am.
Okay, so actually this is a very generalized way of explaining this, that it will control the GFR, but actually there is a very complicated mechanism involved here. So that we'll be discussing later. So how the other processes, actually the blood flow, glomerular blood flow is increased with another factor. The other protein factors that will increase the heart rate, then only the blood flow will be increased. It is not a very simple thing as it is explained in one line here. So later in this chapter, we'll be discussing details about this process. Okay. So the tubular part, tubular secretion, now comes the tubular secretion. Um,
Madam,
the tubular epithelial cells, different segments of the nephron, they perform this
madam, not audible, madam.
reabsorption part. So what exactly is the reabsorption? The selective reabsorption, I missed one thing. This is our glomerular filtration. Selective reabsorption. Selective reabsorption, we know that selective reabsorption. So why selective reabsorption is happening? Because of the gap 180 liter per day to 1.5 liter per day. Okay, so this is our filtrate, this is our urine. So to cover this gap, we need selective reabsorption. Clear? To cover this gap, we need selective reabsorption. Why? Because 99.5% of the filtrate is not going out as urine. Urine is just 0.5%. So where the rest amount is going, that is going back to bloodstream. So that process is called as selective reabsorption. So the tubular epithelial cells plays major role here. At different locations, they take back substances into the blood vessels. So what are the substances? Water, mineral ions, okay, nutrients, what else goes? Like glucose, amino acids, all those things, they all will be going back into the blood. Whereas nitrogenous waste or any other waste substances that has to be removed, that will be taken into the tubular part, and finally urine will be produced. Right. So during the urine formation, the tubular cells secrete substances like the H plus ion. So tubular secretion. So the tube secretes few substances like the H plus ion, K plus ion. So these are ammonia. These substances, they are secreted by the tubular cells. The tubular secretion is also very important step for urine formation, as it helps for the maintenance of ionic and acid-base balance of the fluid. But it is of more important for the organism who have to retain their water in the body. Clear? Is it clear, children?
Yes, ma'am.
Okay. Am I audible to you all? Okay. Any doubts are there? Any doubts?
No, madam.
Okay, children. Okay. Thank you, children.
Madam, happy Navaratri, madam.
Why your voice is not coming? I don't understand.